All right, so here is anaerobic glycolysis. And in this case, you have control of enzyme. So we have our covalent and our allosteric ways of controlling enzyme activity. You have mass action and you have product inhibition. All can play a role in this. I'm going to have to go through these one at a time, and I'm doing that with three important enzymes. One enzyme being hexokinase, which hydrolyzes ATP to phosphorylate glucose and create glucose 6-phosphate. And here you have glycogen phosphatase, same idea. And finally we have phosphofructokinase. And this one also uses the energy from ATP hydrolysis and converts fructose 6-phosphate to fructose 1,6-bisphosphate. And this then is able to continue down the next series of enzymatic reactions, and this is where you have ATP production, and so there's our ATP inflow to the ATP pool. So first let's look at hexokinase. So here's the flow. It goes from glucose to glucose 6-phosphate. As identified, this enzyme hydrolyzes ATP and uses the inorganic phosphate and the energy released there to rebond and create glucose 6-phosphate. So in this case, concentration of glucose is the high part of the gradient, and if you increase concentrations in glucose, you can get an increase in the rate of this reaction. On the low side, increases in glucose 6-phosphate work through product inhibition. And that's identified here, the effect of glucose 6-phosphate. The other thing we're going to start to talk about now is what we call high versus low energy molecules. And glucose 6-phosphate here we code as a high energy molecule. In other words, this is representing that there's a lot of the substances that represent stored energy bonds. So when this increases, it's a signal that less of this glucose breakdown is required because we have lots of this. And so therefore we have product inhibition of hexokinase. It's the primary way in which hexokinase is controlled. The second enzyme is glycogen phosphorylase. So here's the flow with glycogen phosphorylase. And we have glycogen going to glycogen 6-phosphate. And if we look right here, this is GPhos in an active form and in an inactive form. And it's covalent control of this enzyme that makes it active versus inactive. So remember, covalent means we have to attach a molecule. In this case, it's a phosphate ion, which is commonly how things occur. So whether or not we have more of this depends on the inflow into it relative to the outflow. So it's phosphorylase kinase, which is the enzyme that does this phosphorylation, and its activity then is the conductance for this inflow. On the other side, we have the outflow. And the enzyme here is phosphorylase phosphatase. So this takes the phosphate off and increases the outflow from this. You end up with the inactive form. Now again, I've identified what we would call low energy molecules and high energy molecules here. just so you know, this is, you can see this hopefully, this is the blue color. And the blue color over here. And this is AMP, adenosine monophosphate. So now, why is this important? Well, first of all, after ATP is hydrolyzed to form ADP, you can actually have adenylate kinase catalyze the formation of another ATP from 2-ADP, and you're left with an AMP. So you've taken 2 phosphates off here. So you've lost 2 of these energy bonds. This is why we call it a low energy molecule. This molecule, as it increases in concentration, represents the fact that there has been an increase in the hydrolysis of ATP. In other words, the outflow to ATP has increased. Remember, up here is glucose 6-phosphate. So there's a lot of this here. So now let's take a look at how these molecules impact how much active GPhos there is. So this low energy molecule, the more of it you have, it has a stimulatory effect on phosphorylase kinase activity, and therefore we have increased production of GPhos. On the other side, this high energy molecule, glucose 6-phosphate, we have a lot of it. Stimulates phosphorylase phosphatase, and therefore we have an increased outflow and a reduction of GPhos. So we have a connection between energy consumption and the availability of substrate for the eventual ATP production. And if this isn't being used and accumulates, then there's the recognition that we don't have to create more of it, and we have a reduction in the conductance for this reaction. So now we move down a little bit in glycolysis and we get to phosphofructokinase. And phosphofructokinase uses ATP and creates F16 fructose 1,6 bisphosphate, and that then continues on and results in ATP production with a number of subsequent pathways. Okay, so how is this enzyme that catalyzes this irreversible reaction controlled? Again, we look to what are low energy molecules and what are high energy molecules. So here's the regulated pool. And obviously, if we have a lot of that, we don't need to have a high rate of inflow to that pool. This pathway is along the way to that inflow. And so when you have high energy molecule like ATP, you have a molecule that allosterically inhibits phosphofructokinase. So that means it has a binding site on phosphofructokinase, and when it binds there, it inhibits the binding site for F6P. Another high energy molecule is citrate. You'll see when we get to the tricarboxylic acid cycle why we might call this a high energy molecule. And what that essentially does is enhances the impact of ATP, the allosteric impact of ATP on phosphofructokinase. So both of these act together to inhibit this enzyme and reduce anaerobic glycolysis. On the other side, we have our low energy molecule AMP, and the way AMP works is to inhibit ATP's ability to inhibit the enzyme. In other words, AMP will result in an increase in PFK activity. And remember again where AMP comes from. So now we have to look at this highly complex oxidative phosphorylation metabolic pathway. It's not reversible. There's mechanisms controlling enzymes, there's mass action, and there's product inhibition, control of enzymes. So again, recognizing that ultimately we have to have hydrogen ion flow back through the ATP synthase, and that provides the energy that can then be stored in the phosphate bond. You have ATP. So there's this pumping of hydrogens out to where they can flow back in. And we start off with the creation of pyruvate from carbohydrates. And pyruvate, the energy in pyruvate ultimately ends up in the bond in ATP. And here I'm indicating free fatty acids. Free fatty acids also can enter the mitochondria, and they also, therefore, the energy in free fatty acids ends up in this ATP bond. So how is this all controlled? So the first thing we're going to talk about is how pyruvate and free fatty acid both become acetyl-CoA. And acetyl-CoA is the substrate for the tricarboxylic acid cycle. And the tricarboxylic acid cycle, while it does produce a little bit of ATP, its primary purpose is to create electron donors. That's NADH, and H+ that's produced here. And those electrons are for the electron transport chain. Okay, so how do we get from pyruvate to acetyl-CoA? And how do we get from free fatty acid to acetyl-CoA? So first off is the control of pyruvate dehydrogenase activity. So here is the reaction catalyzed by this enzyme, in which we go from pyruvate on one side and coenzyme A and NAD+, to on the other side acetyl-CoA, the substrate for the tricarboxylic acid cycle. And we have an NADH, H+, and a carbon dioxide also produced here. But primarily what we're interested in is this. So PDH is also an enzyme that